Hello and Assalamualaikum. Good day everyone, we meet again. Welcome to UMTMOOC Open Online Course. I'm Ariza from School of Fisheries and Aquaculture Sciences. In the previous video, I have shown you the way to differentiate between male and female fish. Now that you have understand the differences between sexes of the fish, we will proceed with next topic, which is fish breeding physiology. In this topic, we will focus on biological study of function related to fish reproduction and how does the fish reproduction works. To begin with, I will show you the outline of this topic. The first lesson is processes of gonadogenesis. Gonadogenesis have two parts, oogenesis for female and spermatogenesis for male. The second lesson is on hypothalamic-pituitary-gonadal axis. At the end of the lecture, you will be able to learn biological study of functions related to fish reproduction and the mechanism of fish reproduction. Processes of gametogenesis consist of two parts. The first part is oogenesis. Oogenesis is the formation of a haploid and a mature egg capable of being fertilized and supporting a developing embryo. In oogenesis, there are three phases involved, where the first phase is called duplication phase or mitosis. In duplication phase, primordial germ cell duplicate rapidly through mitosis, forming oogonia and remain as diploid cells. In second phase or growth phase, oogonium grows into primary oocyte and undergoes preparatory phase or pre-vitellogenesis. And final phase is maturation phase or meiosis. In this phase, the oocyte undergoes full vitellogenesis. Primary oocytes undergoes meiotic 1 division into a secondary oocyte and a polar body, each being haploid cells. Secondary oocyte undergoes a meiotic 2 division, resulting in an oocyte and other polar body, whereas the first polar body divides into two polar bodies, resulting in one ovum and three polar bodies. The polar body will eventually degrade. At this stage, oocytes are ready for ovulation. The figure shows the oocyte development in the ovary. The second part of gametogenesis is spermatogenesis. It is a process that results in the formation of a haploid and mature sperm capable of fertilizing a mature egg. Similar as oogenesis, spermatogenesis also have three phases involved, where the first phase is called duplication phase or mitosis. In duplication phase, primordial germ cell duplicate rapidly through mitosis, remain as diploid cells. In second phase or growth phase, spermatogonium undergoes growth development to become primary spermatocyte. And final phase is maturation phase or meiosis. In this phase, primary spermatocyte starts meiotic 1 division into two secondary spermatocytes, each being haploid cells. Secondary spermatocyte undergoes a meiotic 2 division, resulting in two spermatid, and each spermatid undergoes differentiation known as spermatogenesis, resulting in spermatozoa. Secondary spermatocyte undergoes a meiotic 2 division, resulting in two spermatid, and each spermatid undergoes differentiation known as spermiogenesis, resulting in spermatozoa. The figure shows the sperm developed in the testes. Next, I will talk about the hypothalamic-pituitary-gonadal axis in fish. It is the relation between hypothalamus, pituitary and gonad of the fish in the reproductive mechanism. Hypothalamus and pituitary belongs to endocrine system. It is situated in the brain of the fish. The initial function of hypothalamic pituitary gonadal axis comes from the hypothalamus. It is located at the base of the brain and sensitive to environmental signals detected from the sensory receptors. It releases gonadotropin releasing hormone or GnRH, which travels to the pituitary gland. Next, the pituitary will receive GnRH from the hypothalamus and releases gonadotropin hormone into the bloodstream. Pituitary is located from the extent of the hypothalamus. The final chain of hypothalamic pituitary gonadal axis is the gonad. It is the organ that produces gametes, ovary or testes. The gonad receives gonadotropin hormones from the pituitary gland such as follicle-stimulating hormone and luteinizing hormone. And gonad secretes steroid hormones to promote maturation of gametes such as estradiol, testosterone, and maturation-inducing hormone. The gonadotropin hormones consist of follicle-stimulating hormone or gonadotropin hormone 1 and luteinizing hormone or gonadotropin hormone 2. It stimulates the gonad by production of steroid hormone and induces secondary sexual characteristics. It controls maturation and reproductive cycle of spermatogenesis and oogenesis. The gonadotropin hormones excretion is controlled by environmental factors and genetics. The steroid hormones consist of estradiol, testosterone, and maturation-inducing hormone. Estradiol is the female steroid hormone and it stimulates the liver to produce the yolk protein vitellogenin, which is influenced by gonadotropin follicle-stimulating hormone. While testosterone is the male steroid hormone and it stimulates the activation of Sertoli cells to induce spermatogenesis. Finally, the maturation-inducing hormone for both female and male, it stimulates final maturation of oocytes in female and spermatocytes in males. The figure shows the hypothalamus-pituitary-ovary flow in the hypothalamic pituitary gonadal axis, where it starts with environmental stimuli. Next, the stimuli will receive by sensory receptor and transferred to hypothalamus. Hypothalamus will secrete GnRH and will receive by the pituitary. Pituitary will secrete FSH for vitellogenesis and LH or final oocyte maturation. Let's talk about the oocyte development. It starts with follicle development, followed by yolk vesicle formation, vitellogenesis, envelope formation, maturation, and finally the ovulation. The figure shows the hypothalamus-pituitary-testes flow in the hypothalamic pituitary gonadal axis, where it starts with environmental stimuli. Next, the stimuli will receive by sensory receptors and transferred to hypothalamus. Hypothalamus will secrete GnRH and will receive by the pituitary. Pituitary will secrete LH to Leydig cell, and Leydig cell will secrete testosterone to Sertoli cell for spermatogenesis, while FSH will directly involve with Sertoli cell for spermatogenesis. Finally, we come to sperm development. It starts with mitosis proliferative, followed by meiosis, spermatogenesis, and spermiation. We are now comes to the end of this topic. As the conclusion, you are now able to understand the biological study of functions related to fish reproduction and finally to study the mechanism of fish reproduction. Hope you enjoyed the video and see you next time. Bye!